Welcome to my video on the adapter hypothesis. Before I get into what exactly the adapter hypothesis is, I will first discuss translation because the adapter hypothesis is based on what happens in translation. And here we have a picture of translation. And to summarize, translation is the process of turning RNA into polypeptides. I would also like to go over the basic information that is crucial to understand for later in the video. So the ribosome is the site at which translation occurs. Messenger RNA is what carries the genetic code to the ribosomes. The transfer RNA is what helps to code an mRNA sequence into a protein, and amino acids are what form the building blocks of proteins. Here we have a really simple visual just to get the point across, and I did not include a polypeptide chain, but if I had included it, it would be over the amino acid. And so basically the adapter hypothesis proposed that small RNA molecules would be adapters that could receive a certain amino acid and have an interaction with a specific messenger RNA. These adapters would subsequently engage in constructing the amino acids into a growing polypeptide. As these adapters are constructing amino acids, moving them along, they are known as transfer RNA molecules. Something to notice here is that the tRNA molecule is anticodons, which are complementary to the codons present on the mRNA molecule. So this is a zoomed in diagram of one CUG codon and the corresponding GAC anticodon that came and settled in on the codon. So the tRNA with the GAC anticodon will have the leucine amino acid attached. This is the way all amino acids are brought on through codons. The polypeptide keeps growing as more codons and corresponding anticodons attach at the ribosome, which again is the site of translation. Now we will compare this headphone adapter for an iPhone to a tRNA molecule. If it weren't for the headphone adapter, there is no way that your wired headphones could connect to your iPhone X because there is no headphone port. Just like if the tRNA molecules don't attach to the mRNA complex, then amino acids can't be added to a polypeptide chain. So in summary, you need a headphone adapter to play music with your wired headphones, and you need a tRNA molecule to allow the polypeptide chain to keep growing. This is why Francis Crick used the adapter hypothesis to propose the function of tRNA in the cell. And lastly, let's say there is an alteration in which there are modified tRNA molecules that can no longer decode stop codons, such as UGA or UAA. Typically, tRNA can recognize stop codons and end the polypeptide chain when it is supposed to, as seen in this visual. However, without the ability to recognize stop codons, as seen in this diagram, the polypeptide chain would keep growing and potentially cause cancerous growth in the cells. This would likely run the cells into overdrive, causing it to either stop functioning or function incorrectly. This could be detrimental to an organism. And here are the references I used. Thanks for watching.